The focus of this virtual lecture is membrane structure and function. Now, when we talk about membranes, we often focus on the plasma membrane, and as we learned, the plasma membrane regulates what gets into and out of a cell. Recall, however, that there are membranes located throughout a eukaryotic cell. We have the nuclear envelope, for example. We have the mitochondrial membranes, both the inner and the outer membrane. We have membranes surrounding the ER. We have membranes surrounding the Golgi. So the eukaryotic cell is full of these membranes. Now I'm going to focus on the plasma membrane as our example. But please be aware when I discuss membranes, I'm talking about membranes in general, using plasma membrane as our model. Now, membranes have many functions in the cell. The first and foremost is the selective uptake of ions and molecules, also the selective export of ions and molecules. So if you really think about it, membranes control the chemical makeup of cells. So for example, calcium is very low inside of the cytoplasm of, for example, a muscle cell. The reason being is because calcium is pumped into the endoplasmic reticulum. And it's the membrane surrounding the ER that controls the uptake of this calcium. Plasma membrane is another example. Right? The plasma membrane regulates what gets into and out of a cell. So a membrane is very important in controlling the chemical makeup of a cell. Secondly, as we just discussed in the previous slide, membranes compartmentalize a cell. If you look inside of a plant cell or an animal cell, we see many of these unique structures that are all separated from the cytoplasm by a membrane. And it's within these compartments that specific functions take place. So if you think about it, where does energy get harvested in a cell? It occurs in the mitochondria. Where is DNA stored in the cell? It's stored in the nucleus. Where are proteins synthesized? Proteins meant for secretion or proteins meant for other membranes, they're synthesized in the rough ER. And what allows that compartmentalization of a cell? It's the membranes that make up these organelles. Next, protein sorting. We've already seen that in the endomembrane system. How can we traffic proteins through a cell? We do so through the use of membranes. If you recall, proteins that are produced in the endomembrane system never contact the cytoplasm. Why do these proteins never contact the cytoplasm? Because they're always surrounded in some kind of membrane-bound structure, including little bubbles of membranes called vesicles. Next function of the membrane, to anchor the cytoskeleton. Recall the cytoskeleton aids in maintaining cell shape, gives a cell tensile strength. It also is involved in transport. For example, transport of vesicles occurs using the cytoskeleton. How does the cytoskeleton maintain its place in the cell? It's anchored to membranes. Next, membranes often contain enzymes. And these enzymes are important in producing energy intermediates that we'll learn about in Chapter 8, like ATP, NADPH. Another good example would be FADH2. All three cases use membranes, specifically proteins in a membrane, in order to produce these energy intermediates for the cell. Cell signaling uses membranes. Now, why is cell signaling important? We're going to talk more about this in Chapter 7. But if you think about it, how can the inside of a cell communicate with its environment? We use membranes. What cell signaling is we have a signal that's located in the environment. That signal binds to a membrane protein found in the plasma membrane. And that membrane protein then relays the signal to the cytoplasm. And now in the cytoplasm, the cell adapts by producing proteins, by modifying existing proteins, etc., etc. So it's very important, if you think about it, that the membrane separates the inside and the outside of a cell. So based on its structure and location, it would make sense that it's sort of the go-between between the environment and the cytoplasm. Membranes play a role in cell division. Lastly, cells play a role in adhering cells to one another. So, for example, liver cells are not ubiquitous throughout the human body. Liver cells are located where other liver cells are, concentrated in that tissue, that organ. Why? Because of adhesion moderated by membrane proteins. Now, in order to understand the function of membranes, we have to understand the structure. Now, the major molecule found in membranes is called a phospholipid. Now, phospholipids belong to the third major class of macromolecules called lipids. Okay. Now, what's unique about phospholipids are their unique character. If you look at a phospholipids, it's actually very different. Has very different character, located all within a single molecule. It has both hydrophilic and hydrophobic character. We call this property amphipathic. So molecules that are amphipathic have both polar and non-polar groups. In this example, let's look at the polar end of a phospholipid. Now, on any quiz or exam, you're not responsible to draw every individual atom or element here in the molecule. What we're going to do is you're going to draw it shorthand. So, for example, here's the polar head group. Now, the reason they call it a polar head group is we can see we have some carbonyl functional groups, which we know are polar and hydrophilic. We have a phosphate group, which we know is polar and hydrophilic. We also have this positively charged nitrogen, which we know would be able to interact with water. So if you look at the polar head group, it's hydrophilic based on the functional groups present. Now let's look at the non-polar end of a phospholipid. Now if you recall, a triglyceride, lipids, a fat, has 3 fatty acids. If you go back and look in your textbook. A phospholipid is a modified lipid that contains only 2 fatty acids. That third fatty acid was replaced by this polar head group. Now let's take a look at those 2 fatty acid chains. Here we see the 2 most abundant atoms here in these fatty acid chains are carbon and hydrogen. Now if I asked you to describe these chains, what term would you use to describe them? That's right. These chains are hydrocarbons. Now let's look at the covalent bond that links hydrogen and carbon. What kind of covalent bond would this be? That's right, that's a non-polar covalent bond. So based on the fact that they're going to share electrons equally in this non-polar covalent bond, what's the partial charge going to be on this hydrogen? That's right, I love that trick question. Recall, there are no partial charges due to the fact that the electrons are being shared equally. So because of the abundance of carbon and hydrogen, we have all of these non-polar covalent bonds, making those tails hydrophobic in nature. So now you can see the amphipathic character that I'm talking about. You have these polar hydrophilic head group, and then you have these 2 hydrophobic fatty acid tails. Now, before I advance the slide here, let's take a quick look at the arrangement of phospholipids in a membrane. Here's a theoretical membrane here I'm going to draw. And I'm just going to draw 4 phospholipids arranged in a linear fashion. And let's say this is the cytosol, and let's say this is the extracellular side. Now, would this arrangement of phospholipids work here in the plasma membrane? Or is there something wrong with that arrangement? That's right, there's no way these phospholipids can arrange in a linear fashion. The reason is this. If you look at the cytosol, a cytosol is made mostly of what molecule? Right, cytosol is made mostly of water. The extracellular environment is made mostly of what? That's right, water. Now, if you look at this arrangement, certainly the polar head groups are happy because our polar head groups are hydrophilic in nature. But now if you look at these fatty acid tails, they are hydrophobic. Therefore, they're going to want to bury themselves away from the cytosol. So we can't organize a membrane as a linear chain of phospholipids. So can we do this? Let me erase that. Will this work? Here's the cytosol. Here's the extracellular environment. Will that work? Again, the answer is no, because the extracellular side is water-based. So how can I arrange, in this simple case, how can I arrange 4 phospholipids in order to satisfy both the polar head groups and the non-polar fatty acid tails? How about I arrange them in this fashion? Again, here's my cytosol. Here's my extracellular matrix. Does that arrangement of phospholipids work? Absolutely, right? Because here we have our polar head groups facing the exterior of the membrane, both the extracellular face and the cytosolic face. And now you have these non-polar tails burying themselves in the center. So what we've created, rather than a single layer, is we've created a bilayer of phospholipids when we construct a membrane. Now the reason I've gone through that is because that's really all the basics we talked about in the chemistry chapter. We talked about the fact that polar molecules like to interact with other polar molecules. There's no better example than water. We talked about the fact that non-polar groups, hydrophobic groups, like to bury themselves from water. We discussed that when we talked about amino acids being buried in the center of a protein. We've now seen the example here with these non-polar hydrophobic, let me just quickly erase that, non-polar. Now we've seen that here in the case of these phospholipids, where these fatty acid non-polar tails are going to shield themselves from water and going to interact through hydrophobic interactions. So now, let's take a look, a little bit more detail. Here's our Phospholipid Bi-layer. We see it takes this shape to protect hydrophobic tails. And it's this bilayer that are a major component of all membranes. So we can see here's our hydrophobic core in the center of that bilayer. And here are our hydrophilic heads. Now we really have 2 leaflets. Each layer is called a leaflet. And this is very important. Each layer is going to be constructed differently. We have an extracellular leaflet or layer. We have a cytosolic leaflet or layer. Now the reason that they are distinguishable from one another is because the extracellular leaflet is going to be in charge of communicating with the environment. Therefore, in order to carry out this communication, the extracellular leaflet is going to have a little bit different chemical composition compared to the cytosolic leaflet. Now, we often call the phospholipid bilayer, specifically when talking about the plasma membrane, we often call it a mosaic model. The reason being is because other than phospholipids, there are many other molecules that we find in this phospholipid bilayer. Here are our phospholipids. Here's that polar head. Here's our tails here. In addition, we also see the presence of another lipid called cholesterol. Cholesterol is an example of a steroid. If you recall, a steroid has a unique structure compared to other fats in that sense that steroids have fused rings, of which there are 4. Okay. So in animal cells we find cholesterol. In fungal cells, for example, we find ergosterol. Both are examples of steroids that contain these 4 fused rings. So that's a second molecule, a second lipid you find in a membrane. I also want to point out these structures here in purple. We have membrane proteins. We have integral membrane proteins. You can see integral membrane proteins span both the extracellular and the cytosolic leaflets. So these proteins actually traverse the entire membrane. We often call them transmembrane proteins. We also have these other membrane proteins called peripheral membrane proteins. You can see peripheral membrane proteins in this case are on the cytosolic side of the leaflet. Here's another example of a peripheral membrane protein that happens to be on the extracellular leaflet. So not only do we have lipids within a membrane, we also have proteins that are located either within a membrane or associated with a membrane. Now another major class of macromolecule we have associated with membranes is a carbohydrate. Now let's look at this example. Here we have a transmembrane or integral protein. On the cytosolic side we have a peripheral membrane protein attached. On the extracellular side we have a carbohydrate attached. Now recall in lab that carbohydrates tend to take a ring structure. Here we could see a series of 5 carbohydrate rings linked together. And this protein that contains those carbohydrates is called a glycoprotein. Now recall, where was this glycoprotein added to the protein, and where was the glycoprotein modified? That's right. When we start to think about where sugars are added to protein right in the rough ER. Those sugars are further modified in the Golgi apparatus. And when the vesicle fuses with the plasma membrane, and that protein is inserted into that plasma membrane, we see that the sugars are always found on the extracellular side of the cell. We have both glycoproteins and we have glycolipids that were made in the smooth ER. We are going to learn the function of carbohydrates are to act as recognition molecules. It's those carbohydrates that allow a cell to identify itself. Either identify itself as a pancreatic cell, it identifies itself as a liver cell, it identifies itself as being my cell. Right? So for example, why do we have different blood types? Because there are different sugars on the surface of our red blood cells. So in that case, we know the sugars help identify my blood type. So we can see a membrane is just not about the phospholipids, although the phospholipids are extremely important. We also have other molecules that play a role in membrane function. Now, how are phospholipids created in the cell? I just want you to have appreciation for this. You don't have to memorize the name of every enzyme. But I want you to have appreciation of, in this case, is that these phospholipids are actually modified fatty acids that are inserted into the rough ER. So the reality is the source of all phospholipids within a cell is the rough ER. And it's the rough ER then that shuttles the phospholipids to other locations through, for example, the endomembrane system. So if you really think about it, the phospholipids that surround a cell originated from the rough ER through this process. So here we see, here's the glycerol that we're going to use. It's an activated glycerol phosphate. We're going to add fatty acids to these hydroxyl groups using dehydration synthesis. Once we've created now these phospholipids, here's the polar head group. That phospholipid's inserted then into the membrane. Once it's inserted into the membrane, that phosphate group is removed, and now what we have is a phospholipid in its final state. Now, how are these phospholipids transferred through the cell? I already talked about the endomembrane system being one way in which this happens. We can see here's the rough ER where phospholipids are produced. Here are vesicles which butt off of the rough ER, which then travel to the Golgi apparatus. There are the new phospholipids to be transferred. Once the Golgi apparatus begins to mature and the cis phase starts to be moved through, we see that vesicles can then break off and carry those phospholipids to other parts of the cell, in this case, the plasma membrane. We also see here that the rough ER can also build up the nuclear envelope in a similar fashion. In this case, we have just lateral diffusion, in which those phospholipids leave and diffuse to the nuclear envelope. We can see here, in this case, that diffusion occurs because the rough ER and the nuclear envelope are continuous. Lastly, we also have proteins that help exchange phospholipids. In this example, the smooth ER is going to transfer phospholipids through a small transport protein. That protein is then going to dock and deliver phospholipids to other organelles that are not in the endomembrane system, like the mitochondria. So now again, what level of detail do we need to understand? We don't have to be able to regurgitate every pathway. What I do want you to know, though, is that the ER is the source of phospholipids for a cell. And that one of the major systems which we did study, which was the endomembrane system, is one of the ways which these phospholipids are transferred. Now let's talk a little bit more detail now about these membrane-associated proteins. As I talked about before, there are multiple types. We already discussed transmembrane proteins. These are proteins that traverse the entire membrane. So we have part of it here on the cytosolic side. We have part of the protein on the extracellular side. We also have these peripheral membrane proteins, which tend to be on the cytosolic side. They often associate with other proteins. They may have important activities, such as enzymatic activities. We also have a third type of protein called a lipid-anchored protein. This is a protein that's been modified that includes a lipid tail. This lipid tail allows a protein to be inserted into one leaflet. We can see here that a lipid-anchored protein, for example, an example of this we'll see later on is a G protein. This lipid-anchored protein, while attached to the membrane, is actually not in the membrane. It actually uses a modified lipid to insert itself. And these proteins are going to play a role in transport, as we'll see. They're going to play a role in cell recognition when they contain those carbohydrate groups on the end. Now, in order to understand how they get inserted and how they function, again, we have to understand their structures. Now, we've talked about this. Membrane proteins originate from the rough ER. And these membrane proteins, once they're inserted into the membrane of the rough ER, are transported in vesicles throughout the cell. Here's a nice picture I took from another textbook that shows that. Here's the ER. Here's my transmembrane protein. We could see it's, let me take out a different color pen here. We could see here, up in the corner, here's our membrane protein. It's been synthesized inside the membrane of the rough ER. We can see that it's been modified on the lumen side with the sugar. We know that a vesicle will butt off from the ER. That vesicle will then transport that protein here, that transmembrane protein, to the Golgi. Now you notice the sugar is always located in the lumen. That's going to be important later on when I build my extracellular leaflet. So now once the vesicle fuses with the Golgi, we can see here that the protein is still in the membrane. We still have the sugar in the lumen here of the Golgi. That sugar is going to be modified as the Golgi matures here to the trans phase. Once it reaches the trans phase, has been modified appropriately, that vesicle is going to bud off. We can see it's still located in the membrane here of the vesicle. We can see that sugar's been modified. And now when that vesicle reaches the plasma membrane, that vesicle is going to be continuous. It's going to fuse with the plasma membrane, become one with. And now when it becomes one with, we see that the protein is now part of the plasma membrane. And we see the sugar that faced the lumen is now that same sugar that faces the extracellular side that plays a role in recognition. So if you really look at it, anything that faces the lumen is going to end up in the extracellular side. And any part that faces the cytosol is going to end up in the cytosolic side of the membrane. So if you look at it, when we synthesize membrane proteins, we use vesicles. If you really look at it and then even more with a more fine eye, you can see that the phospholipids located in the plasma membrane also originated from the endoplasmic reticulum. Because now if you look at step 4, we're expanding the membrane through vesicle fusion. And now we see, because sugars face the extracellular side, we have cell surface recognition. This is important, right? Liver cells are distinguished from skin cells because there are different sugars on the surface of those cells. So that gives you an idea of how we can integrate Chapter 5, cells, to Chapter 6, membranes. Now when we look closely at the structure of these transmembrane proteins, what common secondary protein structure do I see here on the left? Now I'm going to change my pen color again. I'm going to go back to red. Here's 1, 2, 3, 4, 5, 6, 7. Okay. Here's even an 8th example of a specific type of secondary structure. What is that? That's right, that's an alpha helix. So what you can see is the alpha helix is inserted into the membrane. Part of that alpha helix is going to be on the outside. Part of it's going to be facing the cytosol. But even more importantly, part of it's going to be embedded in the plasma membrane. So this is the extracellular side. This is the cytosolic side. Here's the membrane core of the transmembrane protein. Now if you think about it, what types of amino acids have to be abundant on the surface of this part of the protein, or of this part of the alpha helix? That's right. On that part of the alpha helix, we have to have hydrophobic and if I could just change my ink color again, we have to have hydrophobic non-polar amino acids, R groups, facing the membrane. Now wait a second, Professor Weber. You told us that non-polar R groups are going to be buried on the inside. Now you're telling me that these non-polar R groups are going to be on the surface of this particular part of the protein? The answer is yes. I've always been consistent. If you look at the environment here, these fatty acid tails are non-polar in nature. Therefore, non-polar amino acids are going to be able to interact on the surface of this specific part of the protein. Therefore, what type of amino acids are going to line this channel? What type of amino acids are going to be buried in this part of the protein? That's right. Your polar amino acids are going to be buried. Your acidic amino acids are going to be buried. Your basic amino acids are going to be buried. For the simple fact that they can't interact with the non-polar core of this membrane. So now, non-polar amino acids are going to line the hydrophobic surface. Hydrophilic amino acids are going to line this inner channel. Now, the reason that's important, we'll see later on, is one of the roles of these protein is to regulate transport into and out of a cell.